So this is week 3, right? We're in week 3. So if you look on the syllabus, next week will be the first lecture exam. And then it also is talking about the lab exam due dates. So I wanted to give you a heads up on that. So what's going to happen is the lab quiz, and I put that in quotes because it's really an open book, open note assignment. And so that's going to open up this Friday at 1 through your EDUCAT lab page. So your lab instructors have been aware, we talked about this just this morning. So they're going to set that up for you and probably give you an announcement to let you know as well. And then what's going to happen is you'll have until next Wednesday to do that lab assignment. But it's a hard cutoff, 11 a.m. Okay, hard cutoff. So don't forget, put it on your calendar or your to-do list, because this is easy point, all open book, open note. Okay. And so once that closes next Wednesday, in Wednesday's lecture, I'll go over aspects of it because some of it is going to be useful for what you're learning in lecture. And so that's why we're having you do the lab assignment or quiz first, because that's hopefully going to get you better prepared for the lecture exam that will be next Friday, not this Friday of this week. It's going to be week 4, right? So that's the rhythm we're going to be in for the semester. You'll take the lab exams first. I'll talk about them in lecture, and then that following Friday, you'll take the exam. Okay? So I just wanted to give you a heads up on that. So we're already in week 3. Hard to believe. So questions on this idea? Okay, so don't forget, please put it on your calendar or on your to-do list. Hopefully you have some way of reminding yourself. Hey, do you ever do the Google on my phone? Like, hey, Google, and then you ask it to set up a reminder. Have you tried that? Works pretty well. And so sometimes typically I have my phone on me no matter where I'm going. So if I get like stopped in the hallway and someone says, hey, could you blah, 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 blah. And I'm like, oh yeah, but I don't have my laptop with me to like jot it down or a pen and paper. I'll just tell, hey, Google, remind me to blah, blah, blah. And then it does. So that has been really good for me, because sometimes when I bump into somebody and they ask me to do something, by the time I'm back at my office, I'm on to something else and I forget. So that's a lot of stuff going on. So try to think of ways to keep everything organized, especially if you have a lot of classes, you probably work, you got things going on. So it can be a lot, I know. Okay. So this should be available now in EDUCAT, Propagation and Biotechnology. So, boy, I could go on and on with this whole idea. In lab tomorrow, you'll go to the greenhouse and you'll get to pick a plant to propagate. And so you'll get to go through this experience, and they'll talk to you more about the specifics of like how to actually do it tomorrow in lab. But I wanted to give you a little bit of a background before you walk in the lab tomorrow. And so here you're looking at a potato field on the top left there. That's one of the potato fields I worked in when I was a postdoc doing potato scab research. Imagine those fields, that's just this tiny section. The fields would just extend like football field size. We'd have multiple fields with different research experimental designs set up. Well, the potatoes, they're underground. So usually you have a machine come along and harvest those, but because we were measuring potato scab disease, which is on the surface of the tuber, we couldn't have the machine come along and harvest it because the risk of them damaging the surface where we couldn't assess disease was too high. So on harvest day, we would all get out there, and it was usually hot and humid and buggy, sunny. I don't do well with sun, I get burned. And you're down on your hands and knees digging up those potato plants, every single tuber by hand. I had laughed because right after I got my PhD, the very first thing I did when I got hired in that July, and then late July, the first harvest, that was the very first thing I had to do. It was very humbling. You know, here I have my PhD thinking, ah, I got my PhD. And then she's like, yeah, go dig up potatoes. So she came with though, my postdoc advisor. She was always willing to get in the thick of it. So, but yeah, so these tubers that you're seeing on the right, and maybe you've seen this if you've purchased potatoes and you let them sit too long and they start to sprout like that. Okay, so this is another thing I had to learn when I was a postdoc. They talk about seed potatoes. Like my postdoc advisor said, yeah, we need to order some seed potatoes for the next experiment. Seed potatoes? I didn't know what that meant. But basically, there's growers out there that grow potatoes to harvest the tubers, and then they make sure they're disease free, and then they sell you tubers, and those are the seed potatoes because we don't grow plants from the actual seed from the flower. We take the tubers, as long as they have, um, the eye, people refer to it as the eyes on the potato. That's a node region. Remember, tubers are underground modified stem tissue. So there's node region, internode region. So as long as there's a node region, then everywhere you can see it sprouting, that's a successful node area that's sprouting out a new shoot and eventually leaves. And so we would just make sure that every tuber we had at least one eye on it or node, and then we plant it into the soil and it would grow. So it's pretty easy to grow potatoes out. Most of the potatoes you buy in the store, by the way, they've been sprayed to not sprout, but eventually they will. You know, as they get old enough, they will. Or yeah, they age essentially. So, and you should keep them in the dark too, by the way. You don't want to eat green potatoes. Have you ever noticed green potatoes? Don't eat green potatoes. So if the tuber gets exposed to sunlight for too long, the tissue will actually turn green. And that's a chemical being produced called insulin, and it's toxic. You shouldn't, I mean, it won't kill you, but it's not good to eat it. And so I've cut into tubers before where the flesh is pretty green, and you got to just pitch it. Okay, so be aware of that. So that's one way to propagate, is just taking some, cut these up, and then just cut the tubers up. So as long as each cut section has a node, you're good to go. So from one tuber, you can actually get many, many plants from that one tuber. And then this lower image down low on the right is just looking at some stem cuttings, which is primarily what you're going to do tomorrow. You'll most likely take stem cuttings. You might do some leaf propagation as well, depending on what's available. But here you can see that they've cut off some stems from the mother plant, and then they put them into some water. And that's what you'll do, because I want you to see what's going to happen next, okay, when you take that cutting. And so we're thinking about this idea of, you're taking a mature plant, you're not working with the seed, you're taking the mature plant, so it's just vegetative tissue, and you'll take a chunk of that off of a mother plant, so to speak, and then you can get the missing parts to regenerate. So plants are amazing that way. I mean, we can't do that. If our finger gets cut off, we're pretty much done unless they can sew it back on, right? So plants are amazing that way. So what you're going to be doing tomorrow in lab is you're going to look for plants that you can propagate, and you'll be trying to look for the axillary bud. That's a good way to search for a good cutting. If you look for an axillary bud and then you cut below that and plant that into the soil, and you'll start to get root formation and eventually new branches and leaves. And so it's pretty amazing how flexible they are in that regard. You can amputate large parts of them, and they're like, oh, okay, I'll go ahead and do what I need to do. So basically, when you look at the stem cutting at the base of where the stem is cut, let me show you this picture. So where there, you can see those tiny little roots coming off. And what's going on here is that you have right where that stem was cut, those cells right there at the wound area, we say they have to go through differentiation. And I can't fit it all on one line. Sorry. Dedifferentiation. Dedifferentiation. And so this is the idea where basically that's the fancy word for those stem cells right there at the base of the stem have to go, oh, I can't be a stem cell anymore. I need to shift and become root cells, root tissue. So those stem cells are going to go through dedifferentiation, stop being stem tissue, stem cells, and they're going to start to morph into root tissue and root cells. And so they talk about dedifferentiation, and then once they become roots, that's the redifferentiation idea. All right, so that's a phenomenal switch in their gene expression patterns in those cells. And like I said, we can't do that. We don't have that ability. So stem cuttings, typically you take, the more younger growth, which is the growth higher up on the plant, but that's usually works pretty well because they're vigorously growing. And depending on the plant, you might be able to get several sections taken off that stem tissue and create several different plants from it. You just generally you're looking for axillary buds. As long as each cut you have an axillary bud there, then you have a really good chance of this happening. And then typically you will remove the lower leaves because you're going to stick that cut area into the soil. You don't want that the leaves kind of getting in the way of that. So, however, we're not going to put it into the soil. I'd like you to put it into jars of water so that you can watch the roots forming, because that's half the fun of it. There will be a couple of cases where either Duncan or Grant are going to tell you, yeah, let's put it into soil, but it depends on the plant. So this is another strategy that you can do on that lower left image are leaves taken from a relative of an African violet plant. And they just take the leaf off of the mother plant and then bury the end of the leaf. So the leaf, the end of the leaf is buried into the soil, and you see new leaves growing from that leaf that was plucked off of the mother plant. And so that's another strategy that you can do. That works quite well with succulents as well. So that's a good strategy. Sometimes you can take the leaf and the petiole, which is the little stem that the leaf is attached to. So sometimes you can take that and take the blade and the petiole, plant the petiole into the soil and have it grow. So African violets, if you have any African violets in the greenhouse, that might be a good strategy as well. So here you're looking at some succulents up top there where you just take a succulent leaf and bury the tip, the end of it into the soil, and see the little baby leaves starting to form coming off of that. And then more leaf examples on the lower bottom left there. You can see some young leaves growing out. I've never tried this one, the one on the right here. What they've done here is they've taken a leaf and laid it flat on the soil, and then they made incisions into the main vein. And so wherever you see the new leaf is growing, they made like a cross cut here across the vein, and then they laid it down on the soil and kept it moist. And you can see the new growth there. And then the bottom one, that bottom image is, it can often refer to as the snake plant. It grows long, slender leaves. It's a nice, it's an easy plant to maintain in your home. Like I had two of them in my home, and they handle dim light really nicely, and they grow slowly, and so they're pretty easy to maintain, but propagating, you have to be really patient. So the ones that I have at home, I propagated, where you can cut the long length of the leaf and then put the cut end into the soil. So if you do a snake plant tomorrow, you'll probably put it right into soil. And then you have to wait and wait and wait to be patient. And finally, it'll start forming roots, and then it'll start forming new leaf tissue. And the ones that I have at home, they started out like that. And by the time they were almost all weed, I donated them just recently to the greenhouse because they were getting too big for my house. So they are easy to grow. You just have to have some patience. Okay, so those are kind of like standard methods of propagation. And again, you're taking different areas of the plant body and they're pretty flexible. You can get them and encourage them to grow new plants. You can do fancier stuff like tip layering or simple layering, where you can easily take advantage of that by burying parts of them in the soil and get new plantlets that way. And so that's can be a common strategy. I'm just trying to show you some options out there. Okay, so you're going to be doing primarily stem and leaf propagation, but there are other options that people will do depending on what they're trying to do. The mound layering is another one where you mound up the soil. That's kind of fun because it ends forming in the buried tissue like that, and then you can cut them off the plant and plant them. Air layering, that's another one. I've tried that a few times. Again, you have to be a little bit patient for that one, but essentially you're covering up the tissue to get root formation. Lots of options out there. So, I mean, it points to the great flexibility that plants have in terms of their ability to alter their gene expression and start shifting their growth patterns and their morphology so that they can regenerate back into an entire plant. And so another technique that's used quite a bit in industry is tissue culture. And so tissue culture is another way. They often refer to this as micro propagation because it's very small. You start out with little chunks of tissue usually. So here you're looking at a banana tissue culture operation where they're taking cells of the banana plant and putting it onto this agar medium, this tissue culture medium, that has the right ingredients and hormones for those cells to grow. And then you encourage them to differentiate into a full, leaves, stems, roots by shifting the hormone concentration inside the medium. And so here they have kind of a progression going on where initially there's not a whole lot of roots. There's just a little bit of shoots forming, and then gradually you get more and more of the entire plant. Finally, by the down here, it's just all planted into soil. So this is big business. And again, this is all doable because of that totipotency idea that the plants have the ability, as long as you take some living cells from that mother plant, if you put them into these tissue culture situations and you know the right recipes, you can get them to regenerate a brand new plant. It's a clone. It's genetically identical to the plant you started with, which can be good. So if you have like the perfect plant and you're like, oh, I love this plant, I just want to make more copies of it because I could really, enjoy it or make it a profit off of this, you can do this kind of technique quite readily as long as you understand a little bit about the plant and what its needs are to get it to go through the tissue culture process. So there's different recipes out there depending on your plant. And that's something that you might need to troubleshoot a little bit. If you're working with a plant that's not really been done before, you might have to experiment a little. But it's amazing that you could take a mature plant that, is pretty much mature and all of its cells are good to go. And you are taking some of those cells and essentially telling it to revert out and start to be a baby again in a way. It's really quite amazing how you can convince these mature cells that have, finished growing and have their, I'm a leaf cell. And all of a sudden you say, nope, not anymore. I'm putting you in tissue culture and you have to form a brand new plant. Okay. And so I've done tissue culture with leaf tissue, stem tissue, root tissue, and it's all worked. Sometimes it's failed, and I'll tell you a little bit more about that. But one of the more successful ones that is just like, it's pretty reliable is actually working with carrot taproots. And so when I first started teaching plant years ago, that's what we started with. Worked every single time. And one of the reasons it worked so well is that what you're doing is So I would go and buy the fattest carrots I could find, and then we would surface sterilize the taproot. And so you'd have to soak it in, do like a water wash for quite a while. Then you have to soak it in ethanol, and then you do bleach treatment, and sterile water treatment, and then you break it into sections. We actually would use a very small like soil pourer sampler, corkboard, and you would pour into the tissue, not in the dead center because that had too much xylem, and remember xylem's dead, so you can't grab xylem. It's not going to work for you. And so you have to go a little off center, take a core sample, and then you just lay the core sample down in small section and plant it onto the tissue culture medium and let it go. Yeah, so I'll get into that. But basically, when you do tissue culture, one of the reasons that experiment can fail is that you get too many microbes coming along with the tissue, and then they overwhelm the plant. Because this is such an artificial, you're putting them on very artificial medium. It's very easy for the microbes that come along with the plant to kind of take over, and then the plant tissue ends up dying. It gets too out of balance. So you have to do this surface sterilization technique first to get most of the microbes, hopefully all the microbes, on the surface. And then one of the huge reasons why carrot capsules is a good choice is taking internal tissue. And internal tissue is usually, especially if you go internal enough, it's very few microbes in there. And so then you are really enhancing your chance of being contaminant free when you go in and place them. So like I said, this usually worked as long as the surface sterilization got rid of most of the surface microbes, so they didn't accidentally fall into the plate and start growing. Microbes grow so much quicker that they can just overwhelm the plant tissue. So that was very, very easy to do. And so it's a taproot. As long as you get mostly living cells, you're going to be good to go. And so basically what we're talking about is you have pieces of tissue, and you have to make sure that there's no contaminants or pathogens. And then you have to use sterile conditions. So we work with, if any of you have taken plant phys or in our new applied plant science class, we did this as well. We work in sterile flow hoods, we call them, that provide a sterile environment so that you have a safe space to work in that's free of microbes. And then the goal is to get this plant tissue to grow new cells. And initially, it kind of just looks like a lump or mass of cells. So the fancy word for that is callus, which I want you to know that term, please. Be able to understand what that means. So when we talk about callus, someone says, oh, look at that callus. What they're referring to is that it's just like a lump or mass of cells. They're plant cells, but you can't really quite tell what they are yet. They don't look like roots or shoots or leaves. And then under the right conditions and the right mixture of hormones, you convince those callus cells to start to differentiate into root, shoot, and leaves. Typically in tissue culture, the order that I see is the roots come last, which is interesting. It's usually the shoot leaves will form, and then eventually the roots come. And so you have to wait for the entire plant before you can put it out into the soil environment. So it depends on the plant, how you're going to form that recipe, what the ratio of hormones will be. Typically, though, it's auxins and cytokinins. That would be the two hormones. And so do I have that? I'll write it here. So, wow, why it disappears? Auxins and cyto. Come on. I don't know why sometimes my writing disappears on the slide. It shows up and then it disappears. Anyone know about that? It's weird. Auxins and cytokinins are the two groups of hormones that we play with to get the right ratio. Auxins are usually first. That's just grow, grow, grow to get the callus. And then usually you either add in cytokinins or you transfer the tissue into fresh medium that has a cytokinin in it. So this can be very good if you're like, I have the perfect plant. I want to make many, many clones of this plant. This would be one way to do it. Micropropagation, you don't use up a lot of space. You have to have good technique, but you can generate huge numbers of plants this way. And like most of the African violet plants that you see for sale in stores, they're done this way. They do huge tissue culture operations in order to get those different African violet plants. And so it really, I've had a student do a tissue culture experiment over the summer, a couple summers ago, and he was taking tissue from all parts of the plant. It was amazing, and he got it to work no matter what tissue, as long as there's living cells. You can't take just xylem, but he was taking living cells from leaf and stem, root tip, apical meristem. He was getting it to grow in all cases. It was really fun. So you have to usually add in other things besides hormones. You usually give some mineral support into the medium. You typically do add sugar because they can't do photosynthesis well enough yet. So you give them some sugar to work with. And then those hormones I mentioned. And then sometimes, depending on the plant species you're working with, they might tell you to add in coconut milk, which is essentially coming from the endosperm tissue area inside the fruit. And then sometimes you'll see stuff like yeast extract they want you to add in. And that often provides some good like B vitamins, things like that. So, like I said, lots of different recipes out there, and you have to do a little bit of research to figure out what recipe would work. But if everything goes okay, like we typically use this, I don't know if you can see from where you're sitting, but these are like little square boxes or rectangular boxes with lids on, clear lids for the oxygen in there. These are called magenta vessels. So that's what we work with. We do tissue culture in our classes. And then you just put the tissue there, and that gives enough headspace for the plant to grow large enough so it's quite vigorous and good to go. You can easily then transfer it into soil right from these vessels. And so, like I said, probably the biggest thing to worry about is contamination from microbes that come along either from the surface of the plant. So let's say you take we've tried African violet tissue culture where we go and buy African violet plants from a store in town. And so they've been out in the real world. Their leaf surface is covered in microbes. If you've ever looked at African violet leaves, they're very hairy, lots of trichomes. And so they're fairly delicate leaves. So that the trick we kept running, or the problem we kept running into, is the surface sterilization techniques either would not be done long enough or strong enough. So we would get contaminants, or we'd overdo it and we'd kill the tissue. So it's like, so it can get a little frustrating depending on the leaves or the plant species, I guess. So you have to be a little careful, but getting it surface sterilized is a big deal. So now in like plant phys, what we do is we start with the preppers before the students get them. We have the preppers work with the seeds because surface sterilizing seeds is pretty straightforward compared to surface sterilizing a leaf, for example. And so if you surface sterilize a seed and then put it into a sterile condition and then grow a baby plantlet and then have the students take that sterile plantlet and work with it, then it works out. But taking plants from the real world and going through this can be a little tricky. So having everything sterile. And so you have to realize this is a very artificial system. And so if there are any microbes lingering, they can quickly create an imbalance where they overgrow the system. And I've seen some really nasty looking vessels within a couple, within a week. You can see that they just swarmed over the agar medium and the plant tissue, and you can tell the plant tissue is not going to make it. So on the other hand, I've seen some, well, I'll get into that idea in a second here. But so we often use the word plantlets, which is in that down low there, it shows the word plantlet. So when they're, new little baby plants, we often just call them plantlets. So that's a phrase you'll hear a lot in the tissue culture. So the contaminant idea, here you're looking at a published paper where they were working with cannabis sativa. and they were looking at how to get rid of all the microbes, like what conditions would allow for that to happen. In this image, you can see that there's not just the plant tissue in these culture beads. So all that white stuff, that's all fungus. Here's huge block of fungus. Here's a funky bacteria growing out. Root tissue and bacteria, fungus. All right, so when it gets this bad, where it's so overwhelming microbial growth, typically the plant tissue will die because the microbes start using the tissue for nutrition. And so surface contamination can be a problem, but if you get the right procedure down, you can deal with it and get a clean tissue. The other thing you have to worry about when you do tissue culture is the microbes inside the tissue. Okay, so just like you have billions of microbes in you, plants do as well. Okay, and so the fancy word for that is endophytes. And endophytes are generally, we define them as microbes that hang out in the plant tissue, inside the plant, not in their cell. Okay, so they're not in the cytoplasm, they're just in the tissue. And we're finding that these endophytes do help the plant. So we have many good microbes in us as well that help us. So for example, right now you have millions and millions of E. coli in your intestines, and they're making vitamin K for you. They can do that, we can't. All right. And so in fact, the minute you were born, the doctor or nurse probably asked permission to give you a vitamin K shot because you don't have enough microbes in you when you're first born to have vitamin K. And so that's one of the first things they do is give you a vitamin K shot. You have to get colonized by E. coli to really get good a dose of vitamin K. So anyways, many plants have been studied. The more we look for endophytes, they're there everywhere, every plant, and they're doing good things for the plant. Well, you can't do internal sterilization of the plant tissue. You're going to kill the plant. All right. So this is a problem. And so endophytes, you can't really get away from. You have to it's a lot of luck and some good technique to try to minimize this happening. So what you're seeing here in this image, this is carrot tissue. And you can see that if you take a core of carrot taproot just under the skin, so to speak, of the taproot, just under the epidermis, sometimes you'll have endophytes. So even in the taproot, the carrot taproot, a few millimeters into that tissue, in that region, you can have endophytes hanging out. And so what you're seeing here is the orange blocks are the carrot tissue. And then you can see this growth coming out of the tissue. All right, so you can see that it's growing out of the tissue. Those are probably endophytes. So what happens is they came with the tissue, you put the tissue down onto the agar medium, and they started to grow out onto the agar. And they like the nutrients provided, so they'll keep growing. Now, there's some other issues going on here too. You see these other colonies of microbes growing on the plate away from the tissue, right? So this is telling me whoever did this prep had microbes that came in and fell onto the plate when they were manipulating. And so this was not in a sterile environment, or somehow microbes got wafted in there, fell on the plate, and started to grow. So there's a couple things going on here. But when you do tissue culture, you always monitor your tissue right away. Like the first 48 hours are kind of critical. You look for anything growing out of the plant. You look for anything that might have fallen in during your manipulation. And so I've rescued tissue, like if I see it like this, sometimes you can rescue if the tissue is in a clean, relatively clean area, like I can probably rescue that. But this would be hard. If you see this endophyte growing out of the tissue, trying to take that tissue out and redo, put it on a fresh media probably wouldn't work. So these endophytes you have to deal with, they're going to be something you always have to think about. One of my grad students he was trying to do tissue culture of rare orchid species out in nature. He was trying to figure out a way to clone orchids by just taking a tiny leaf sample so that people, I don't know if you were aware, but people will like go and hunt orchids and rip them out of where they're living and take them home. It's been a thing since, the 1700s. And we're trying to prevent that, of course. You don't want those going to extinction. So he was thinking of, could we learn a tissue culture just from a tiny leaf sample and clone these plants? He kept running into endophyte issues, though. This was quite a while ago when some of the recipes weren't really out there. And it was a real problem for him in order to try to deal with this. So endophytes can't really be helped. However, probably the best way to try to minimize any endophytes, any surface contaminants, is to actually take apical, shoot apical meristem or root apical meristem tissue. That's probably your best bet. So this is an example out in the real world where the cannabis industry is currently dealing with this hop latent viroid. Have you heard about this? I don't know if any of you in here, maybe the medicinal plant chem professors are aware of this, but the hop latent viroid has jumped into the cannabis plant, and it, started in the hop plant, which is related to cannabis. But the best way to deal with getting rid of this viroid, okay, so this is not even a virus, it's a viroid, which is just a piece, by the way. If you don't know what a viroid is, it's a little piece of RNA, and it's infectious, and it's bizarre. And so viroids are something that can attack and cause disease in plants. We don't know of any viroids that attack humans, but I put in gold highlight there. Meristem tissue culture is the only way to deal with this. And it's the same like with endophytes. If you want to guarantee, I don't want to get any contaminating endophytes in this tissue that I'm going to try to clone, you want to try to get at the meristem tissue culture technique. And so you have to work with that really small cluster of cells at the shoot tip or the root tip, which is challenging. It's probably one of the harder tissue culture processes. You have to be very precise. So they're referring to this viroid as the COVID of cannabis right now because it's so widespread and it's causing all kinds of financial difficulties for the cannabis industry. So it's a big deal. Some of the quotes from some of the reports I was reading, 90% of California cannabis is infected with this viroid, $4 billion in lost yields. 40% of cannabis flowers sold in Canadian dispensaries tested positive. Luckily, it doesn't hurt humans. So yeah, it's a lot. It's a big deal. And so I have a former student, a botany student who's working downstate, Danielle, and she contacted me last fall asking if I knew of anyone doing tissue culture work in cannabis because their grow operation was getting infected by this viroid. And she was trying to get hooked into some business that could provide some tissue where she could do apical meristem tissue culture. So it's a big deal. Okay, so micropropagation or tissue culture techniques. It's the more, as the years have gone on now, there's more and more recipes out there, more techniques have been really like nailed down and kind of perfected. And so this is getting to be bigger and bigger in the commercial industry of growing plants. And so if you get out and get a job in the plant world, chances are you're going to encounter some aspect of this with many of the businesses that you might be looking at for employment. So something to think about. Questions on tissue culture? I assumed that was an annual loss. Yeah, I think that's how they were referring to it. Has anyone heard about this viroid? Yeah. So I don't know. I mean, it sounds like it's going to be quite the challenge to deal with it. But, I know that there are cannabis tissue culture businesses out there. There was a new business that I can't remember the name of it, but one of our former medicinal plant students went and worked for them, Ben, downstate, and he was doing tissue culture work. So before he left for the interview, he asked, hey, can I come into your lab and do some tissue culture work just so I can get a feel for it, do it a little bit before I go for my interview. He did that for about a month or so, and then went and interviewed and got hired. So he was doing tissue culture for that company. So they were like a new company that kind of recognized we need to get into the tissue culture realm of growing these plants. Okay. Let's do a eye clicker.